Phytophthora, that literally means plant destroyer, is a genus of pathogenic oomycetes whose member species are capable of causing enormous economic losses to crops worldwide as well as environmental damage in natural ecosystems. Charles Montaigne, a retired French army doctor, first described the fungus as Botrytis infestans in 1845, and later in 1876 the great German pathologist Heinrich Anton de Bary named it as Phytophthora infestans. Approximately 100 species have been described so far, although 100 to 500 species are believed to exist. So far as the systematic position of this genus is concerned, it belongs to kingdom Mycetae, division Mastigomycotina, subdivision Diplomastigomycotina, class Oomycetes, order Peronosporiales, family Pythiaceae, and genus Phytophthora. The origin of Phytophthora can be traced to a valley in the highlands of central Mexico. The first recorded instances of the disease caused by it were in the United States, in Philadelphia and New York City in early 1843. Winds then spread the spores and it crossed the Atlantic Ocean with a shipment of seed potatoes for Belgian farmers in 1845. It was first introduced in India in the Nilgiris during 1870 to 1880. Species of Phytophthora cause a variety of devastating diseases on many different types of plants, especially dicots ranging from seedlings of annual vegetables or ornamentals to fully developed fruit and forest trees. Most species cause root rots, damping off of seedlings, and rots of lower stem, tubers, and corms. Others cause rots of buds or fruits, and some cause blights of the foliage, young twigs, and fruit. Phytophthora root and stem rots cause damage to their hosts in nearly every part of the world where the soil becomes too wet for the good growth of susceptible plants and the temperature remains fairly low, that is between 15 degrees centigrade to 25 degrees centigrade. The best known species is Phytophthora infestans, the cause of late blight of potatoes and tomatoes, but several other species also cause extremely destructive disease on their hosts. Phytophthora cactorum, Phytophthora cambivora, Phytophthora cinnamoni, Phytophthora citrophthora, Phytophthora frageri, Phytophthora palmivora, and Phytophthora syringae cause primarily roots and lower stem rots, but also some cankers, twig blights, fruit rots of woody ornamentals and of fruit and forest trees, as well as of vegetables and other herbaceous plants. Several other species such as Phytophthora capsicii, Phytophthora cryptogaea, Phytophthora megasperma, Phytophthora parasitica cause root, stem, and fruit rots of many vegetables, ornamentals, and field crops, and some woody plants. In some cases, the oomycete also attacks and causes partial or complete rot of the fruit, as in pepper, cucurbits, tomato, strawberry, citrus, and cocoa. The mycelium of Phytophthora infestans is profusely branched and consists of aseptate, hyaline, cenostic, moderately thick hyphae about 4 to 8 microns in diameter. The hyphal wall is electron transparent, approximately 0.1 micron thick. Glucan is the predominant material in the hyphal wall. Cellulose is a minority component with small amounts of hydroxyproline. The plasma membrane is distinct, undulating, and frequently forms lomosomes. The cytoplasm contains scattered diploid nuclei, dictyosomes, mitochondria, endoplasmic reticulum, ribosomes, and many large vacuoles with lipid inclusions. The fungal hyphae ramify in the intercellular spaces between the cells of the host tissue as intercellular hyphae. There are also hyphae that penetrate, traverse, and eventually leave the cell. These are intracellular or transcellular hyphae. Haustoria develop on intercellular hyphae when the latter come in contact with the primary walls of host mesophyll cells. They are small, globose, short, straight or curved pegs, one or more in each host cell and most common in tubers. Experiments have shown that 21 degrees centigrade is the optimum temperature for mycelial growth, and about 26 degrees Celsius hyphae die within a week. With the onset of favorable conditions during spring, a tuft of slender branching hyphae arise from the internal mycelium. They push their way out through a stoma or epidermal cell on the lower surface of the leaf. On the potato tuber, they appear in large numbers on cut surfaces, but normally push through the lenticels or injured portions of the skin. These aerial hyphae are hyaline, sympodially branched, and are of indeterminate growth. Such spatially branched aerial hyphae are called sporangiophores or conidiophores. Phytophthora thus differs from other members of the family in having sporangiophores distinct from the somatic hypha, which is an indication of specialization. Now, the development of the sporangium. The sporangium is formed by the inflation of the tip of the side branch of the sporangiophore. The multinucleate inflation is then cut off by a transverse septum or basal plug as a sporangium. The hyphal branch bearing the young terminal sporangium continues to grow. As the sporangium reaches maturity, the branch tip swells slightly just below the sporangium and proliferates, pushing the sporangium to the side as the elongation proceeds. This process may be repeated many times. The sporangia are thus born terminally but are subsequently shifted to a lateral position. Temperature and humidity appear to be the two most important factors determining sporangial production. Abundant sporangial formation takes place in culture between 9 and 22 degrees Celsius, but 18 to 22 degrees Celsius is the optimum range, at which sporangia are produced within 14 hours, whereas at lower temperatures, 48 hours are required. As for the relative humidity, 100% is optimum and 91% minimum for sporangial production. The mature sporangium is hyaline, oval to elliptical or lemon-shaped and thin-walled spore sac with a basal plug. It has a small stalk and an apical papilla. The sporangial wall is nearly 0.3 microns thick and appears to be faintly layered. Within the wall is the plasma membrane, closely pressed to sporangial wall and encloses the multinucleate cytoplasm, which contains the usual cell organelles such as mitochondria, dictyosomes, endoplasmic reticulum, and ribosomes. In addition, the cytoplasm of the mature sporangium contains paired basal bodies associated with the nuclei, membrane-bound packets of microtubules, and several types of vacuoles, particularly the cleavage and the flagellar vacuoles. The former originate from the dictyosomes and are randomly distributed in the cytoplasm. The latter are arranged along the sporangial wall. If we analyze the dispersal in Phytophthora, it is found that infection of susceptible plants take place by means of sporangia that are blown by wind, rain splashes, or contact with other leaves, or are transported by water and washed into the soil. Sporangia may be dispersed to the neighboring fields but do not generally survive long distance travel because of desiccation and exposure to solar radiations. Now the sporangial germination. The sporangium germinates either directly by a germ tube that enters through a stoma and infects the leaf, or indirectly by means of zoospores. The method of germination is largely governed by temperature. Low temperatures around 12 degrees centigrade favor zoospore production, that is indirect germination, and higher temperatures germ tube production, that is direct germination. The optimum temperature for direct, that is germ tube germination, is 24 degrees centigrade. Indirect germination takes place in cool and wet environment. Under these conditions, the sporangium behaves as a zoosporangium. During differentiation, the multinucleate sporangial protoplast is divided into 5 to 8 uninucleate daughter protoplasts, each one of which metamorphoses into a biflagellate reniform zoospore. Of the two flagella, one is of whiplash type and the other tensile. The flagella arise from the depression on the concave side. Liberated zoospores swim about actively in a film of water for some time and then come to rest. They retract flagella and may encysst. The encysted zoospores then germinate by putting a special short hypha called aprissorium, from which a minute infection peg grows and enters the epidermal cell of the host. No aprissorium is found if the entry is made through stomata. After penetration into the host tissue, the germ tube develops into a profusely branched intercellular mycelium, sending long curled haustoria into the leaf cells. A few days after infection, if the weather is favorable, numerous sporangiophores emerge from the stomata and give rise to a large number of sporangia, which are spread by wind and infect new plants. A large number of asexual generations are thus produced in one growing season if conditions favor the development of the fungus. I feel it pertinent to mention here that indirect germination of sporangium through the intervention of zoospores is an example of the retention of an ancestral character. Reproduction of zoospores is therefore a primitive feature in its life cycle. In wet, warm environment at higher temperatures from 20 to 24 degrees Celsius, direct germination takes place. Here, the sporangium behaves like a conidium and germinates directly by a germ tube. The emerging germ tube may branch or may not branch at all. Most of the growth takes place at the hyphal tip. Various species of Phytophthora, like Phytophthora palmivora, produce chlamydospores. These develop as oblong intercalary or terminal spores. Most have relatively thick walls, but this varies with the species. In mature chlamydospores, the cytoplasm is dominated by lipid and reserve vacuoles. The first authentic report of the discovery of a sexual stage for Phytophthora was published by Clinton in 1911. Later, experimental works has shown that the fungus is monoecious, self-incompatible, and heterothallic, that is, requires two mating types designated as A1 and A2 for sexual reproduction. The A2 type was originally recognized from Mexican isolates. where it was found to occur in equal proportions to the A1 type. Sexual reproduction takes place by means of antheridia and oogonia of opposite mating types, as they are self-sterile. Antheridium arises as a short lateral hypha from the mycelium, with its tip inflated to form a more or less clavate structure known as young antheridium. It is thin-walled and contains non-vacuolated cytoplasm with a single nucleus or more often two nuclei, and is not separated by a septum from the supporting hypha or stalk. Oogonial incept arises as a short lateral hypha without any inflation. It contains dense multinucleate cytoplasm. While still in the process of development, the antheridium is punctured by the oogonium of opposite mating type, which grows through it and develops into a globose structure, that is, oogonium above the antheridium. The antheridium thus forms a funnel-shaped collar around the base of the mature oogonium. This type of antheridium is termed as amphigynous antheridium. Oogonium has dense multinucleate cytoplasm when young. Towards maturity, it increases in size and the protoplast becomes vacuolated. The spherical nuclei, which are about 40 in number, undergo meiotic divisions. The oogonial protoplast becomes differentiated into an outer or peripheral hyaline zone with vacuolated multinucleate cytoplasm surrounding a central uninucleate region with denser cytoplasm. The former is called periplasm and the later ooplasm. All the nuclei in the periplasm later disintegrate. Prior to fertilization, the single nucleus in the ooplasm divides into two daughter nuclei. One of these degenerates. The surviving one functions as the egg or oosphere. The antheridium is delimited from its supporting hypha by a septum and contains about 12 nuclei. These are formed from the original two nuclei of the young antheridium by division after penetration by the oogonial branch. Prior to fertilization, all the nuclei except one degenerate. The surviving one functions as the male nucleus. During fertilization, the oogonial wall bulges at one point into the antheridium. This point is called receptive spot. It then dissolves, and through the opening, the antheridium pushes the fertilization tube and extends upwards into the oogonium. Within the oogonium, it travels up the oogonial stalk, penetrates the periplasm, and enters the oosphere at the nearest point. Here, it opens up at the tip to deliver the male nucleus and some protoplasm. The fertilized egg secretes a heavy wall around it and becomes an oospore. There is empty space between the oogonial wall and the thick oospore wall. The oospore thus partially fills the oogonial cavity and is described as aplerotic. It is a resting spore and thus considered to be an important overwintering structure which plays a significant role as a propagule in the disease cycle. Fully dormant spore has a thick wall differentiated into 2 or 3 layers. There is a large, dense spherical body in the center with sharply defined edges known as the reserve globule. Surrounded by several spherical globules, lipid or oil in nature, perhaps providing energy for its long dormancy. Germination of oospore takes place after the decay of host tissues and on the onset of conditions suitable for germination. Germination occurs at temperatures ranging from 12 degrees centigrade to 25 degrees centigrade. Oospore absorbs water and swells. The diploid oospore nucleus divides repeatedly. The increase in the number of nuclei is accompanied by the increase in the number of mitochondria. The central globule is surrounded by vacuoles indicating its digestion and absorption. Oospores germinate by means of a germ tube that usually terminates in a sporangium, as in Phytophthora infestans. At times, however, the germ tube will produce mycelium, as in Phytophthora cactorum. The contents of the sporangium may divide to form zoospores. Each, after liberation, germinates to give rise to new mycelium. Where meiosis takes place is not clear, but is expected to occur in the gametangia, and the life cycle is therefore haplobiontic diploid. Phytophthora is an oomycete fungus that causes potato blight or late blight disease of potatoes. It is the culprit behind the devastating Irish potato famine that occurred in mid-18th century. The genus Phytophthora includes pathogens that cause root, stem, foliage, flower and fruit diseases of plants, although the pathogen is generally considered soil-borne. Late blight, despite the use of various control measures, is still regarded as one of the most devastating diseases of arable crops worldwide. The vegetative body is the mycelium, which is profusely branched and consists of aseptate, hyaline, cinostic, moderately thick hyphae, about 4 to 8 microns in diameter. The fungal hyphae ramify in the intercellular spaces between the cells of the host tissue as intercellular hyphae. There are also hyphae that penetrate, traverse and eventually leave the cell. These are intracellular or transcellular hyphae. Haustoria develop on intercellular hyphae when the latter come in contact with primary walls of host mesophyll cells. Asexual reproduction takes place by means of hyaline oval to elliptic thin-walled sporangia borne on spatial reproductive hyphae, the sporangiophores. These sporangia germinate either directly by a germ tube that enters through a stoma and infects the leaf, or indirectly by means of zoospores. Sexual reproduction is oogamous and takes place by means of antheridia and oogonia of opposite mating types. During fertilization, the antheridium delivers the male nucleus and some protoplasm into the oogonial egg. The oospores thus formed germinate by means of a germ tube that usually terminates in a sporangium. At times, however, the germ tube produces a mycelium to begin a fresh cycle.